Our presentation is about the sponge, whose scientific name is Porifera, meaning pore-bearer. Sponges, while they may look like plants, are actually members of the animal kingdom and are classified in the Porifera phylum. There is debate on whether the first sponges emerged in the Precambrian or Cambrian era, but some fossil records date back to approximately 600 million years ago, making them one of the oldest, most primitive groups of animals on Earth. There are between 5,000 and 11,000 species of sponges around the world, and they are mainly marine with about 150 species that live in fresh water. Sponges are the most well-known animals within the Parazoa subkingdom. They are carnivorous invertebrates that are multicellular, heterotrophic, lack cell walls, lack tissues, lack organs, and typically they are asymmetric. Most are sessile, which means that they are firmly attached to the ocean floor, rock, or reef where they live and do not move, though a few are able to slowly crawl. Various species range from under a half inch to 11 feet in length, and they can weigh up to approximately 20 pounds. Sponges can live for thousands of years, with the oldest living animal being the Antarctic glass sponge, with an estimated lifespan of 15,000 years. Each sponge is made up of a skeleton composed of the fiber spongin, a mineral known as silica or calcium carbonate. The skeleton supports a system of canals and pores through which water flows, driven by waving flagella. Structurally, the sponge is studded with numerous pores called ostia that lead to canals for channeling water to internal chambers. Sponges are attached at one end to a hard surface, while the opposite end, called the osculum, remains open to the aquatic surroundings. Sponges have a three-layered body wall consisting of the pinacoderm, which is the outermost single layer of flattened cells called pinacocytes, the mesol, which is a thin jelly-like middle layer similar to connective tissue in animals, is composed of collagen, spicules, and cells called archaeocytes that aid in digestion, nutrient transport, and are even capable of developing into sex cells, and other cells called sclerocytes produce the spicules, which are skeletal elements that provide structural support. Lastly, the choanoderm, which is the inner layer of the body wall, consists of cells called choanocytes. These cells contain a flagellum, which is surrounded by a collar of cytoplasm at its base. Through the beating movement of the flagella, water flow is maintained and directed through the body. Sponges have a body plan with a pore or canal system that is arranged into three types: asconoid, ciconoid, or leuconoid. Asconoid sponges have the simplest organization, consisting of a porous tube shape, an osculum, and an open internal area known as sponge column that is lined with choanocytes. Ciconoid sponges are larger and more complex. They have a thicker body wall and elongated pores that form a simple canal system. Lastly, leuconoid sponges are the most complex and largest of all the three types. They have an intricate canal system with several chambers lined with flagellated choanocytes that direct water flows through and eventually out of the osculum. Sponges are most commonly found on ocean floors or attached to rocks, coral, shells, and marine organisms. The majority of sponges are found in saltwater, though some species like the Spongilla lacustris are found growing under rocks and logs in freshwater lakes and rivers. There are three main classes of sponges, which are Demospongiae or demosponge, Hexactinellida or hexactinellid, and Calcarea. 90% of the world's sponges are found within the class Demosponge, which alone contains 4,750 species. Their size can vary from 3 millimeters to over 2 meters in length. They also vary in shape and color and are found in all parts of the ocean. Hexactinellid sponges, also known as glass sponges, can be found all over the world, but are particularly populous in Antarctic waters. These sponges stand upright and reach heights of up to 30 centimeters, forming strong bases that hold them to the ocean floor, with open central cavities that allow water to pass through. Most hexactinellids exhibit radial symmetry. They may form symbiotic relationship with shrimp, who live inside of the sponge's central cavity. Calcarea are the only sponges whose skeletons are made up of calcium carbonate. This class contains more than 400 species that can be found all over the world, but are most commonly found in temperate waters such as the Indian Ocean, at shallow depths less than 1,000 meters. These tiny sponges grow to less than 10 centimeters tall.
There are two main types of sponges that we use to categorize all the different species. The first of these is known as freestanding. Freestanding sponges have a larger inner volume compared to their surface area. These sponges can grow to be over 6 feet long. The other types of sponges are known as encrusting and are often compared to moss in the way that they can line rocks. Sponges are one of many different aquatic animals that eats through a filter feeding process. Most sponges feed on bacteria and organic material using pores called ostia, which are small openings where water can enter the body. Within these pores are choanocytes, or collar cells. These collar cells surround the flagellum, which creates a current in the water, and the collar cells trap particles inside. Sponges do not have a circulatory or respiratory system. This means they must get their oxygen from within the water. Through the process of diffusion, a sponge pumps water into its body, while nutrient and oxygen-rich water passes over the cells. Oxygen diffuses into the cells, while non-essential particles or waste products, such as carbon dioxide, leave the cells. This process occurs passively because the water moving over the cells has a higher oxygen concentration than the area within the cells. The method of ingestion the cells use is known as phagocytosis. The following video shows an example of how we can visualize the way sponges pump water effectively. A syringe is filled with non-toxic dye called fluorescein and squirted at the base of the sponge. Then it is clear to see how the water is moving as the dye is pumped through the sponges out of the osculum. Sponges can reproduce sexually or asexually. Sexual reproduction occurs through the production of egg and sperm, and individual sponges have the ability to produce both male and female gametes. Fertilization takes place when sperm cells from one sponge are released through the osculum and carried by the water current to another sponge. As this water is propelled through the receiving sponge's body by choanocytes, the sperm is captured and directed to the mesohyl. Eggs reside in the mesohyl and are fertilized when they fuse with a sperm cell. After fertilization, a larva is formed and leaves the sponge body until it settles on a substrate where it becomes attached for the rest of its life. Asexual reproduction is less common, but occurs through the process of budding, in which a new individual grows out of the body of the sponge that either remains attached to or separate from the body of the parent sponge. This process is called fragmentation, in which new sponges develop from sponges that have released fragments of themselves, or through gemmule formation, in which special groups of cells called gemmules can be produced and released to develop into an entirely new sponge.
Sponges have the incredible feature of regeneration. Regeneration is the ability of a new individual to develop from a detached part of another individual. Regeneration also enables sponges to repair and replace damaged or severed body parts. In the following video, we can see the process of regeneration on a cellular level. When the cells are passed through a sieve, they are separated. Sponge cells may be separated by mechanical methods or by chemical methods. The disassociated cells then settle, migrate, and form aggregates in which the archaeocytes play an important role. In order for small clumps of cells to form larger massive cells, the cells must generally become attached to a surface, where they flatten and develop an envelope of pinacocytes. This is called the diamorph stage. Reconstitution of the choanocyte chambers and of the canal system follows soon afterward, resulting in a young sponge that is functional and able to grow. In the natural world, during unfavorable conditions, sponges are reduced to small fragments that may consist of only masses of archaeocytes covered by layers of pinacocytes. A complete sponge forms from these fragments when favorable conditions return.
Human interaction. Sea sponges are very well known as a natural alternative for bathing and cosmetics. So nowadays, a lot of people, nowadays in the U.S., we usually use a synthetic one that's kind of modeled after the animal, but there's still lots of people out there that use actual sponges. It works because they have a relatively soft sponge and skeletal structure that makes them very useful in like cleaning and absorbing water and dirt and bacteria and stuff like that. They have great use to humans in their chemical defense mechanisms. Sponges can defend themselves by producing chemically active compounds. Some of these compounds are antibiotics that prevent pathogenic bacterial infections, which is actually something I'm going to talk about in sponge ecology.
Back in the day before synthetic sponges became popularized, sponges were harvested by hard hat divers with a rake-like tool to pluck the sponges from the ocean floor. Bags of sponges were lifted to the boat above, and the crew cleaned the sponges, which were then hung to air dry. After days or weeks in the water, the boats would return to shore where the sponges were auctioned off to the highest bidder, trimmed, sorted, and then sold to the public.
So sponge ecology, their sponges are pretty critical for the ecosystem of coral reefs. They provide shelter for a lot of organisms like shrimp, crabs, and algae. Um, they provide protection and food supply. So yeah, it's very, very important for the coral reefs. Sponges can really affect the water quality of the reef because they can remove up to 95% of the bacteria and particles from the water. So really healthy for and very important for the food web. Fun fact: sponges actually share 70% of the human genes. So be very surprised how similar we are to sponges. And we're excited. Thank you.